I would like to introduce our first guest speaker, Eric Kuhn from Chimera Therapeutics. So Eric is Associate Director for Targeted Proteomics at Chimera. I think people who have been in mass spec field for a while certainly knows Eric's name from different papers. So Eric has spent more than 16 years at the Broad Institute developing assays for multiple large collaborations and consortium, for example, CPTAC. And before that, Eric has spent more than 20 years in the pharmaceutical industry, being responsible for analytical development and biomarker discovery. So without further ado, I'd like to hand it over to Eric and wish everyone enjoying breakfast and a great presentation.
Wonderful.
Thank you. I'll change it down.
Thank you, Yuhan. Good morning, everyone. Thank you for coming out early on a Monday morning. Very excited to share. But first, I'd like to just thank Biognosys in general, Jakob, Yuhan, Oliver, Roland, for the invitation and the opportunity to talk to you today about advances in approaches for using target protein mass spectrometry, target mass spec, to measure pharmacodynamic effects of target protein degradation in the clinic. So just a quick disclaimer. I'll start just by introducing the TPD, the target protein modality, how at Chimera, quantitative proteomics has really enabled the development of drug TPD degraders throughout the discovery and drug discovery pipeline, highlighting, which button we have here? Highlighting a paper that came out late last year in Nature Medicine, where we were able to show in patients with atopic dermatitis and hidradenitis suppurativa, deep and sustained degradation of IRAK4 in PBMCs from blood and skin using targeted mass spec. And before that, really going back into some of the pre-analytical, assay development that was, the advantage of a degrader versus an inhibitor. Other particular focuses on sample preparation so that everything in the assay was ready to go before that first in-human study. And then finally, there are conclusions, primarily that the assay is fit for purpose to do exploratory measurements of target protein degradation, and that we're still working on challenges such as PBMC stability, so we can really include multi-center trials and dealing with recovery and methods for extraction from tissues that are very heterogeneous, like skin. So at Chimera, we're really inventing new medicines, harnessing the power of the ubiquitin proteasome system. The ubiquitin proteasome recycles, removes unfolded, denatured, aged proteins using enzymatic cleavages that it sort of gets, the indicator there, recycled back. In TPD, we're taking a small molecule that binds to the protein of interest, tethering it together close to an E3 ligase in the presence of an E2 ligase that allows ubiquitin tags to be placed on that protein, and as it's earmarked, gets shuttled then into the proteasome and degraded. And this process is actually catalytic. This degrader can go back and repeat the process over and over in the cell. And so this functionality actually opens up more of the proteome to be drugged. Compared for some of our molecules, IRAK4, MDM2, TYK2 inhibitors don't get full inhibition or don't sustain the inhibition for periods of time. And with a heterobifunctional degrader, you can actually remove the protein altogether. In other cases, there are undrugged targets, STAT3, STAT6, that the pocket is not accessible for inhibition. And there you can just also remove the protein altogether, either with a bifunctional degrader or also with molecular glues. And then finally, there's a lot of protein expression that's varied across tissues. And so picking a different E3 may actually enable to target different tissues or remove the liabilities associated with a pan-drug therapy where toxicity may really prevent it from being used in the clinic. And so at Chimera, we have two molecules that are in phase 1 studies for oncology. We have STAT3 in liquid and solid tumors and an MDM2 degrader for other indications. We also have two molecules that are about to enter phase 1, one for STAT6 and one for TYK2 with multiple different indications. Today, I'm just going to really focus on our lead molecule, KT-474, an IRAK4 degrader that we have developed in partnership with Sanofi. And so with the continued advancement of proteomics technologies, the instrumentation, the software, the hardware, the computational tools, and so on, that increase the depth and breadth of the proteome that can be measured and quantified by mass spectrometry, including post-translational modifications, medicinal chemists continue to develop drugs to remove, modulate, and change the levels of one protein. And so there is still a great opportunity to have assays that can measure one or two proteins with really deep, sensitive, and reproducible measurements. Biomarker assays, whether it's proteomics or in other assays in general, are typically expected to measure proteins that go up in concentration. In targeted protein degradation, we're measuring things that decrease by 90% or greater. And so assays that can be linear down to 1 order lower or 2 in cases where the protein is in much lower concentration of the cells really are an advantage. In addition, the clinical timelines, sorry, let me take a step back. I mean, there are multiple enrichment strategies to improve the sensitivity of detection, whether it's antibody enrichment or fractionation or other ways, but those take time to develop and qualify. And so looking for a method that can directly measure the protein of interest on a nanoscale that can be transferred to CROs supporting the clinic is really imperative to keep the assay relevant relative to PK and the other assays such as ELISA on that clinical timescale. And so here is an overview of how we use quantitative proteomics at Chimera to qualify and develop drugs and degraders. We really start with a discovery experiment, whether it's DIA, TMT, offline fractionation, deep dive, thousands, 8,000, 9,000, 10,000 proteins where we're looking for potential off-targets, as well as potential biomarkers. From there, we will take those peptides and proteins and develop stable isotope labeled peptides, LC-PRM, and direct measurement assays that will allow us to move into in vivo studies in mouse, rat, dog, monkey, rabbit, pig, where we have peptides specific for each species, where we can then measure the degradation in cells and also in tissues, heart, liver, kidney, lung, spleen, and especially skin, which is really a challenge. And eventually those assays can then be translated and made robust and qualified for use in the clinic, which is what we're doing right now for STAT6 and TYK2, getting ready for those first-in-human studies. So our first-in-class degrader, I'll delve more into degrading IRAK4 and the implications for biology in the clinic. IRAK4 is sort of a master regulator of innate immunity. You know, tethered with MyD88, it will take input from toll-like receptor agonists as well as interleukin-1 receptor cytokine input and start the either in a catalytic function or in scaffolding pro-inflammatory inflammation, Th1/2/17 immune response. And compared to an inhibitor, shown here with a readout of interleukin-6 after stimulation, the degrader is more potent, it's much more left-shifted, and it's a more sustained reduction of that cytokine blocking inflammation, really showing that there is an advantage to removing the protein altogether. In addition, we've done phosphoproteome-wide studies to show the reduction of downstream kinase activity and verified this more in efficacy models, in preclinical efficacy models. And so assays were developed to measure IRAK4, both a flow assay shown here on the left and antibody-based, where you can measure IRAK4 degradation across a number of cell types. as well as calibration curves shown here in PBMCs at diagnosis, linear range, getting back to that aforementioned linear range of 1 or 2 orders deep degradation we can measure with these assays. The antibody assay, in contrast, has got this background noise that really prevents measuring really sustained based on the noise of the antibodies and the fluorescence readout in the system. But also collecting patient blood over periods of time and intervals, there are limits to how much you can draw and when you can draw it and how you're going to process it. And so we developed a method that allows us to get from a single draw of blood both plasma for the PK and small molecule quantitation and PBMCs or PD with targeted mass spec. We also measured with both peptides sort of the relative stability in the first 4 hours, which is the range in which most of this isolation would take place. And then looking at in a dose curve in PBMCs with this degrader, you can see single-digit nanomolar degradation, a very potent IRAK4 degradation by both peptides. And then also showing similar levels of quant, whether we're doing it in skin, which is much lower than in PBMCs. This is sort of just an approach of the study that was used in healthy volunteers, showing both proof of mechanism by flow and mass spec, as well as biology with an ex vivo stimulation measuring the cytokines in plasma. On the other side, skin was also collected for similar proof. IF was done, staining to show degradation of IRAK4, mass spec, and then RNA-seq, which I'm not going to present here today. The trial design itself, there was 2 phases. The first parts A and B, A being a single dose study with a number of cohorts, and then also a multiple ascending dose with daily dosing out to 14 days across a number of doses shown there. Part C was really the part of the second phase of the study, went out to 28 open label, 28 daily doses. It was open labeling in patients with HS and AD. And of course, with all phase 1 studies, the primary readout is safety and tolerability. But it's also an opportunity as an exploratory to measure the knockdown of IRAK4 and PBMCs in the skin. Shown here is figure 1 for that Nature Medicine paper. On the left is the absolute quantitation of femtomole per microgram IRAK4, coming down to a nadir around 7 days for most all doses. And that's very close to the limits of quantitation here in the assay, and it continued out to day 14. This is in the MAD portion of the study, the 14-day daily dosing. And then you see sort of a dose-dependent rebound after out to 28 days. On the right is how we plot a lot of these data relative to the pre-dose, showing deep degradation greater than 90%, greater than 95%, both at day 7 and day 14 for all the doses in the study that were statistically significant there. But also in skin, we were able to get good exposure from an oral dose degrader, much higher than we saw in plasma. And based on this dosing, if you compare, this is for AD and HS, the pre-dose compared to the levels of IRAK4 measured by targeted mass spectrometry, they return to be similar to those healthy volunteers in the MAD study. So that indicates that we're engaging the target, we're reducing it back to levels, and we're actually seeing a decrease in those elevated levels in those 2 indications. So in conclusion, between the SAD and MAD studies, between the oral dosing, there was deep, robust, sustained degradation of IRAK4, both in the single and the multiple dose studies. It was well tolerated. And then in the patient cohorts, we saw similar degradation, and we even saw some very promising preclinical activity based on this. And based on this study and based on these results, the assay and the phase 2 study was then transferred to Sanofi, and that's going on right now. And this is just more indication of sort of a reduction here in the HS patients in addition to the degradation. But that's not the only project we're working on. We also have Iraqimid, where we've been able to decrease IRAK4 by mass spec, and Icaros and Ilos. STAT3 degradation for our STAT3 program. And we also have other assays for the MDM2 program as well to show a pharmacodynamic effect of those degraders. So we're very encouraged, very excited about all this progress. In short, the assay is fit for purpose for measuring that, but it was really based on all the background, the foundation, and the framework that was developed before we got to the clinic, whether that was just the cross-species validation, how we handled the samples so that the assay was ready, and then we also had mechanisms in place to move this over and translate the assay across sites. There's still challenges that we are pursuing. PBM stability, that 0 to 4 hours, we would like to extend that. That will allow us to process more samples, bring in multi-centers, have a bigger, expansive trial. And then, as I alluded to earlier, getting more reproducible extraction efficiency of our targets out of a very heterogeneous tissue such as skin, especially a skin that's being divided in many ways, just like the plasma and the blood draws for different readouts, so we can use all these together. And hopefully I've shown you today that we're really using target mass spectrometry as a suite of assays, whether it's flow, IHC, ELISA, to really comprehensively understand the pharmacodynamic effects of our degraders. And with that, I would like to thank you for your attention. Happy to answer any questions. One of the answers to this question is it does get sunny in Boston, just not today. So the sun does shine, but not on the day we had our summer outing. Thank you. Yes. That's a great question. As an exploratory assay, we looked at some stability, we looked at calibration curves. We did not do a full validation in the sense that this is an endpoint in any trial. In fact, as you go to phase 2 trials, they'll probably not likely use a lot of this readout. They've already shown that they can get degradation. So we're really qualifying these assays with stability, reproducibility, precision, sensitivity, and more in that line as a qualification for an exploratory readout, not the final endpoint within the trials themselves. Thanks. Thank you. Yes? Yeah, I think there's still a niche. That's a great question. We're still in the process of using DIA a lot for our in vitro studies, in the selectivity and off targets to verify those DC50s and Dmax by DIA, and can we get the same result by PRM? I think translating a DIA method into the clinic is going to be that's a longer road to hoe, where right now we're going to still do that transition so we can get 20 scans under the peak, more precise quantitation, and have a number. This number, percent degradation, I can go to it too much. But a lot of times they don't have an efficacy readout, cytokine or others. So showing you can reach 90% degradation or greater is actually going to be an efficacy readout. And so having more precision around that number allows the doctors and everyone to make a call on how well we're engaging the target. So yeah, DIA has come along and helped us a lot with the selection of peptides for targets and building the assay, but eventually it has to transfer into an in vivo setting, complex tissues, PBMCs, where we need that rigor, and I think that will continue as we move forward with those assays in the clinic. Yeah.
Yep. Thank you.
Yep, thanks.
Thank you, Eric.